welcome back to the lecture series in animal cell culture or cell culture technologies. So, we are into the third week and we have finished the first 3 lectures of the third week. So, today we are entering into the fourth lecture. So, as of now, if you look at it, we have extensively talked about how to lay the foundation stone of a facility. Specifically, what all possibilities. I may have missed out certain things, so please feel free, add up on the existing layout of things. But I believe that I have pretty much covered most of it, what is needed in the modern day cell culture facilities. Till in future, things are going to move towards more of a microfabrication lab on a chip. So, that is one section. Later half of the course, we are going to kind of discuss couple of classes, how things are changing, how there are smaller versions of incubators and laminar flow hoods and all those kind of things which are popping up in the market. There are a lot of R&Ds going on across the world. So, today, after giving you this overall outline of how you can set up a lab, what we will do after this virtual lab, we'll enter to that virtual world where we'll be talking about the dishes or the culture vessels and the series of culture vessels and what you have to keep in mind, how you have to keep in mind, what are the prerequisites and what all measures you have to take for swift functioning of your research. So, those of you have done a little bit of a cell culture or have worked in any of the bioengineering or biology or chemical engineering labs or material science lab, as a matter of fact, where they work a little bit on our biology side, you must have seen some form of vessels. Okay, they call it T70, T20 flask and likewise. So, just to give you an overview, what all the different shapes of vessels you must have seen. So, let us start today's class. We are into the week 3 and lecture 4, W3 L4. Okay. Now, some of the dishes you must have seen something like this. Okay, 15 mm dishes, transparent dishes of polycarbonate, very thin polycarbonate. You must have seen this. You have a cap. Okay, you have the dimension around 15 centimeter, and the height will be around, you can say for example, I am going to be able to tell you the exact, something like this, something like 1 digit, what will be the height? Okay, something like this much is the height. Okay, so you can call it like this much, and you have a lid. Now, what you do is the cells are growing on this base. Out here the cell grows and you fill it with medium halfway through. You cannot fill a lot of medium because you do not want the cells to submerge in such a way that they are deprived of oxygen. So, now this is one kind of dishes you must have seen. Then you must have seen 90 mm dishes. This is 15 mm dishes. Then you have 90 mm dishes which are bigger like this with a lid like this. Then you have something called a T20 flask. You must have seen these flasks which are, from the side view of the flask, looks like this. Okay, and the cells grows here, and again this is how far you fill the medium, and these flasks comes in different sizes. There are a bigger version of these flasks, just the same way as this kind of dishes, a bigger version, smaller version of dishes and flask. Sometime in a very old textbook, if you some of you referred, but such things are no more existing, or most of the lab doesn't do it anymore. It was something which was followed in the very early days in National Institute of Health, United States at Maryland. So they had this very interesting concept. They used to have a, instead of this polycarbonate, that was a very similar stuff of glass. You can get them fabricated, very thin glass. The reason for some of these to be made up of glass was exactly the same reason what I mentioned in one of the very recently previous classes, what I told you that numerical aperture of the thickness of the material is very critical when you are visualizing with the microscope, numerical aperture of the objective. So, earlier when there were not very many objectives which could be used for the polycarbonate, they used to have this glass stuff, and then followed by there is something in between which came. So, if this was glass and these were all polycarbonate, PC, I am just representing by PC. In between there was something which is even much more funny. So, they used to have this polycarbonate stuff like this, and there used to be a small, they used to cut a small hole on the bottom, and top of that they used to put a glass cover slip. So, essentially most of the cell growth was happening on top of that glass cover slip, and that cut used to help to visualize it using a microscope, because that cut was good enough to get a complete field view. Or they used to cut it slightly bigger, like, you know, say for example this cut having this big. Okay. So, these are, if you say, more or less the kind of vessels which will you come across in any cell culture lab. But what is important here is the part 2 of this, the science what we are going to deal with. And one more thing, you will see people can directly grow cells on the base of these dishes, on the base of these dishes, on the glass cover slips. So, you have these glass cover slips of different dimensions. You can get very thin glass cover slips which are very suitable substrate, okay, and you can grow the cells on top of glass cover slips. And similarly, you have the exactly the same thing, 15 mm dishes or 20 mm dishes of glass, and you have this T20, T80 flask. Now, first thing what is important for you to realize, what kind of vessel you are going to use. So, in order to address this problem, there are two different kind of questions you have to ask. Question 1, cell type. Cell type means, are we talking about adhering cells or non-adhering cells? So, if we are talking about non-adhering cells, so it means we are talking about these cells. Say for example, I have a vessel like this, and I have the medium filled with it. This is the medium, and I have these cells coming in. So, they are growing on suspension. This is called suspension culture. If it is a suspension culture, it really does not matter, because it does not require any kind of base matrix to grow. It is independent of the base matrix. Okay, these non-adhering are independent of base matrix. Fair enough. Now, at this stage, we are not talking about these ones because these are a different class of cells what we deal with. If we talk about the adhering cells which needed a base matrix, and this base matrix means extracellular matrix or ECM. So, if your cell type needs an ECM, then you have to figure a way around this. What are the ECM molecules which you will be needing? Because there are two options here again. Okay, let's, before I get into the detail, either your adhering cells grow directly on polycarbonate or directly on, of course this is also polycarbonate, or directly on glass cover slips or directly on that surface. If these cells have some way or other, so the way it works is very simple, let me tell you. So, say for example, let us pick up a color. So, green color will represent the cell. Okay. So, these are the cells what you have to plate. These now, just say for example thousands of cells what you are going to plate on a substrate. So, here is your substrate. This is the dish where you are vessel where you are going to plate the cells. So, what will happen? So, what you do with a small amount of medium, so very small amount, so that you allow these cells, you plate these cells on it, on this culture dish. On the culture dish at the base you have small, that little bit of a medium, right, totally very little, okay, which will kind of form a layer on top of it, and the cells will be rolling on it like this. And you literally can see under the microscope how wonderfully the cells will be rolling. Many of them may try to, so you have to keep the base parallel to the surface. It has to be at 180 degrees so that on in either side it should not tilt. Okay. Now, this is at time t0 as soon as you have plated the cells. They look like this. And of course, there are lateral mobilities which are happening, faster, slower, depending on how gently you are plating them. But there will be certain degree of mobility in all directions they will have. And eventually, you will see, it's funny to see them under the microscope, eventually they settle down at some spot. Now, the first thing these cells does, now I am introducing a red color, and that's, so say for example, assume, so our assumption is, so my assumption is cells will adhere or attached to the polycarbonate or / glass surface. That can only happen when these cells on their surface, they have some kind of a charge, okay, may have a positive charge, negative charge, whatever. Now, say for example, on surface they have certain degree of positive charge, and the polycarbonate or the glass what you are having is having some form of a negative charge on its surface. So every material has certain degree of charge on its surface. It's a very static charge, but there will be certain degree of charge on the surface. Now, the positive charge of the cell surface, so the interaction is something like this. Okay. So here you have the cell positively charged on the surface. And this positive, okay, you might be wondering from where it is coming. So you have all these glycoproteins, several other positively charged ions which are kind of embedded on them. So just for the timing, assume that it has certain degree of positively charged molecular charge, or vice versa. You can assume the other way, that it has a lot of negatively charged. So then you have to think the other way. So at this point, I am assuming that your cell surface is positively charged. Okay. See, the point is positive, negative. It's just we give a symbol, but just oppositely charged, which will attract, and your surface has, say, negatively charged. So there will be electrostatic interaction between these charge moieties, which will ensure the initial, how to put it like this, the initial weak linkage is forming there. There won't be a very strong linkage. It will be, kind of something like this, slightly flimsy, but there will be I won't say very, kind of will adhere very fast, but there will be something which they will be kind of, at this zone they will be kind of, rocking like this. Okay, that will be the first set of interactions which will happen. Now, post this interaction, something else is going to happen. What is that something else? Something else is, upon these interactions, these cells started to secrete certain protein or certain matrix molecule, which I am showing in the pink color. Those are called the name is self-explanatory, extracellular matrix material. Extracellular matrix material. These are material which are composed of different kind of proteins and carbohydrates and series of long chain molecules, whose role is something like this. Say, for example, this is your cell. This is your cell. Okay. Now, and here is the substrate, and here you have the first set of interaction with the substrate, with the positively and negatively charges, where you have the first set of interaction which will help them to kind of settle down at a spot for a while. Now, this extracellular stuff is something like this. Something like this. So, it is kind of anchoring to the surface. These are anchoring molecules. Depending on, I am just kind of extended it for your understanding. And very interestingly, almost, this is one experiment I did somewhere around, if I remember, somewhere around 2003 or 2002, I remember, yeah, maybe 2001, and that we did a very funny experiment. We just plated the cell like this. So, we plated the cell, and by a very brute force method, after plating, after say 2 hours, after 6 hours, after 10 hours, we removed the cell from the surface. Okay. What we observed, and after we removed the cell from the surface, so what we are left with, essentially, if you see this picture, we are then, if you could remove the cell from the surface without destroying its surrounding environment. So, you have extracellular matrix, which is possibly or probably is deposited on the surface. Now, in this situation, if you have antibodies against this extracellular matrix protein. So, for those who are not aware of what I am telling you about in terms of extracellular the antibodies are, just imagine I have a specific molecule, there is a binder which is very specific to that molecule. Okay. So, those are called as antibodies, just for a very layman understanding for you. Okay. If I have an antibody against these, just putting at ABs, okay, then what we observed is almost within, this is how low we have gone. I think we managed to went all the way up to 1 hour or so soon after plating. You see there are traces of extracellular matrix deposited, which otherwise the surface we never had anything except polycarbonate. So, that means this first set of interaction which is happening out here between the positive and negative triggers a signal to the nucleus of this cell where the DNA resides and tells the DNA, dude, I need these extracellular proteins, extracellular matrix protein to be synthesized and anchor myself because I want to settle down here, because we are dating with this surface now. Okay. So, it is something like this. So, think of it, one small electrostatic interactions could really trigger the DNA to play its role, and that starts a very unique way of looking at the whole problem from the perspective of surface biochemistry or surface cellular biology, surface cell chemistry or cell biology, whichever way you prefer to use the word. And this is something which is very critical. You might wonder where this is all going to go. This is something critical to figure out why certain cells in our body. And okay, before I tell this, let me add one more interesting aspect to this. Now, this nature of this kind of molecules which are formed, extracellular matrix, is unique for cell type to cell type in terms of quantity and quality. That means, say for example, there are set of, say I say that just for your understanding sake, I say there are 5 different kind of ECM complexes which are formed. Now, for each cell type, it is not essential that they all will be using all the 5 types. Some may use 2, some may use 3, some may use 5. But now, if suppose you have a situation where 2 cells uses all the 5, then 2 you can distinguish because they may use differentially. They may secrete 1 larger as compared to 2, 3, 4, 5. They may secrete 2 and 3 larger as compared to 1, 4, 5. So, you can have an n number of combinations. But that brings us to a very interesting point. Extracellular matrix are cell specific. Okay. Depending on the cell type, they secrete that unique form of extracellular matrix. Now, you have to ponder, you have to think over it before I continue this story. In the next class, what does that mean and what is the implication of understanding this interaction? Okay. I will close in here. Thank you. Thank for your patient listening. We will continue this next class.